Hello, everyone. Good afternoon. Welcome to this afternoon session where we have three different talks presented by three different speakers from different nations. And this is the last session of this incredible workshop. It's dedicated to wildlife applications. But first of all, let me thank Licia Colli and Paolo Aimone Marsan for having invited me to co-chair this session. So, Licia.
Yeah. So we start by inviting the first speaker of the afternoon session, Xavier Dallaire from Université Laval in Canada, who will talk about Arctic charts.
Hi, thank you for having me. It's been great to meet all of you and hear more about your work. I'm Xavier. I'm a PhD student in Jean-Sébastien Moore's lab at Université Laval, working closely with Louis Bernatchez in his lab, so in Quebec City, Canada. So we've heard a lot about projects in Italy and other regions of the world, and I'll take you to a completely other corner of the world, which is the Canadian Arctic. Also, I'll talk mostly about a species important for fisheries. So especially in southern Canada with all the fisheries on Atlantic salmon, but also Pacific salmon, there's a lot of use of genomics to resolve stock structure, to inform management, also know more about the adaptation to environment of different populations, and also identify key populations for conservation. But what about the Arctic? So the Arctic is a really remote area where not a lot of research has been conducted. So my project tries to fill that gap and provide genomic information on a key species in the Arctic, which is the Arctic char. It's in the family of salmons and trout, so the Salmonidae. It is the most harvested fish species in the Inuit Nunangat, which is this big territory in red that you see, which is mostly inhabited by really small Inuit communities. We're talking fly-in only communities, so no road network. So they are very isolated and very dependent on hunting and fishing because the groceries are so expensive. So Arctic char is a species we find all around the globe, all around the pole, actually. It has an outstanding diversity of morphs. You can see different morphs, and you can see how the one I circled in is the anadromous form, the one that goes from the river to the sea. And you can see why it's the most harvested one, just because it's so much larger than morphs that stays in the lakes. So it is a really important species for alimentation, subsistence, and there's not a huge concern for the species as a whole, but there has been stock decline in the rivers nearest to communities. So that spawns hatchery and stocking projects all over the Inuit Nunangat. And of course, we know that there are really rapid environmental changes in the Arctic, even more so than the other region of the world. So because of all of this, we think it's a especially interesting species to study. So when I talk about anadromous, I said that it's the migration pattern of being born in the river and going in the sea to feed and grow. So anadromous salmonids are some of the most studied wild animals in landscape genomics, just because they're so important for economical reasons. So we know that a lot of those species are highly phylopatric, so they go back to the river where they are born to reproduce. So that creates reproductive isolation and really strong genetic structure, and can lead to local adaptation, of course. But study on local adaptation in salmonids have mostly focused on the freshwater habitats, just because different populations tend to occupy the same marine habitats. So maybe there's less of a chance for divergent selection to occur. However, in Arctic char, things are a bit different. So because it's so much to the north, the summers are really short, and it's going back every year, every winter in the river, which is not the case for most salmonids, who can stay multiple years at sea. And we observed that they do tend to stay much closer to the coast and near the river mouth where they are born. So there are more potential for local adaptation of Arctic char, both to the freshwater, but also to saltwater. And also when working in the Arctic and a lot of other places in the world, it's hard to ignore the glacial history of the region. So you can see on the little animation just the last glacial maximum that covered most of North America. And it's really, really recent in an evolutionary scale. And it's obviously been affecting genetic diversity in numerous taxa. Mostly because while during the glacial maximum, we can have the ranges of species drastically reduced and survive only in some refugium. And that can lead to demographic bottlenecks and lineage divergence. And then when the glaciers, the ice recedes, you can have, you'll have recolonization, dispersion, and different lineages will have to adapt to a changing environment. There's numerous example on that point. And most of the time we see mostly glacial refugium happening in the south to the ice border. But in the Arctic, there are evidence from different Arctic studies, Arctic species on mitochondrial DNA that suggests that there could have been some glacial refugia in the high Arctic. And Banks Island that you can see here, and that in some maps on the ice extent was completely free of ice even during the maximum, have been proposed as a candidate for the location of a refugium. As far as Arctic char phylogeography, what we know of, it's split even in North America in multiple glacial lineage. The blue dots that you see in the middle are what we call the Arctic lineage, and is what I'll be mostly focused on today. But you also have the Atlantic lineage that come actually from Europe, and that we find in the Alps also in yellow. And we have the Bering lineage more to the west, Russia and Alaska. And there is a contact zone of the Arctic and Atlantic lineage that was observed in on the Labrador coast in eastern Arctic. So that leads me to my research interest. So I'm very interested in studying how the past, present, and future environment of the Arctic impact populations of Arctic char all across northern Canada. So I'm using both landscape genomics and phylogeography analysis to resolve this. So I'll be presenting you some results on both those subjects. First with landscape genomics, which is mostly part of my master's that we published in 2021, actually. So I'll start with that. So like I said, these are a really remote region. So we were really lucky to have numerous partners. A lot of people study that fish just because it's so ubiquitous in the Arctic and so important. So we had the chance of myself getting some samples either by hiring local guides and fishing by gill nets, or to install sometime with bigger projects, counting fans on rivers to catch the fish in their upstream migration. So when they just leave the sea and go back to their natal river. So you can see my sampling map on the left side, and we try to cover different spatial scales. So we both have also groups of sampling sites separated by a few hundred kilometers, and also really close by sampling sites in neighboring rivers. So we use ddRAD-seq for sequencing and SNP calling, and ended up with around 18,000 markers, NIPS markers, in 650 samples in the 21 sites I've just shown you. So just starting with a general genetic structure with admixture, we can see that using a k = 4, how the different groups of populations are structured, and really fits with the different marine environments. So you can see the Hudson Bay in the west, the Hudson Strait in the Elgava Bay, which are three big bodies of water that are quite contrasted on the environmental standpoint. Like I said, and those groups can be further divided in multiple populations, subpopulations, and I thought it was interesting to see how really close by populations could not be differentiated based on our data set. So we can expect that in those close by river, there might be a lot of gene flow happening. But one interesting case is those two rivers that really are sharing an estuary. They flow in the same bay, the Leaf and Bérard River. And going further into this, we found it really interesting that they actually stand on the border of two major geological provinces, so the Superior Province and the Churchill Province. So the rock underneath is really different. So we think maybe that it can explain the absence of gene flow between those two populations, because a lot of salmonids use their sense of scent to find their way back to the river. So maybe the water chemistry of those rivers might be really different and might prevent gene flow between. We also calculated FSTs and found some strong patterns of isolation by distance, as we expected, along the coast, but also across the Hudson Strait. And we also compared the genetic diversity through heterozygosity in different populations. And we've seen some differences that I'll try to dive in more deeply. And especially in the Hudson Bay, we found a lower genetic diversity and really low differentiation also between the populations. I think that it might be because, and I'll show you soon with the deglaciation maps, but it might have been a really rapid demographic expansion from north to south in Hudson Bay. Also, it's just at the end of the range expansion. There's not really that much populations to the south. So just to show you, so you can see the blue line, which is the extent of ice 12,000 years ago, and progressively moving, and how we think it might have, it can explain the recolonization of the region by the chars that could have come both from the Arctic lineage and the Atlantic from the southeast, like I said earlier about the admixture of the lineages. And gradually, at some point, multiple thousands of years later, the Hudson Bay and Elgava Bay could have been recolonized. So maybe that explains why that fits the expectation that genetic diversity might be lower in most recently recolonized regions because of serial founders effect that can reduce genetic diversity. So next, the main part of this this paper was to detect a gene environment in this region. So this is really a contrasted region, both in so from left to right, you have air temperature, precipitation, sea surface temperature, and then salinity to the far right. So we used those factors, but also many others I'll present later. Using three methods. So we use both RDA and a partial RDA that we talked a lot about yesterday, also a LMFMM method. So as far as the environment goes, so we wanted to modelize both the freshwater and marine environment of the species that is moving a lot, but still staying in some areas that we can define. So for the freshwater factors, we use the watershed that you can see in green. Some are quite big. It's not necessarily clear how far the fish go in those huge watershed that you can see on the map. But most of the time we expect salmonids to be able to complete especially big migrations. And for the saltwater factors, we use a coastal habitat. So we stay between the shore and 5 kilometers offshore, just because we know that are from telemetry, acoustic telemetry studies that they do stay close to the shore. So our first goal was to be able to distinguish signal of natural selection that might be different in freshwater and marine factors. We use factors from WorldClim, BioOracle, and MarSpec that you can see here, different air temperature, precipitation, salinity, the tides, productivity, and so on. But like we talked yesterday, a big problem with GEAs, so Gene Environment Association, is the problem of collinearity. So we had a lot of collinearity. The study area is, like I said, is separated in multiple bodies of water that do co-vary a lot in their environmental factor. So we were extra careful, maybe even too careful, but we reduced those factors to 3 axes of PCA that you can see here. You can see on the plot on the left how each of the factors relate to each of the PCA. So those are the factors that we use in our RDA. So like we saw yesterday, we try to relate the genotype to the environment. And when it is a case of partial RDA, we try to correct for population structure or at least spatial correlation. So I used distance-based Morans eigenvector maps that we touched upon in the different other presentation. So it's just a matter of having a variable that represent how close by the populations are. Since we've seen there's a lot of isolation by distance, you expect if they are close by to be more related in their general genetic material. We'll present the result both for the RDA and then the PRDA. So we were able to, so you'll see a lot of other presentation I've seen is based on the individual. So you have individual genotype. In that case, we work on a population basis. So the input is actually allele frequencies than actual individual genotype. You can see on the left side the outlier markers that I've colored based on which PC they most closely related to. And you can see on the PRDA that we find also a set of markers. And what I find interesting when looking at the R2s, the R squared, is you can actually try to check how much of the genetic variance is explained only by the environment, only by spatial structure, and by both. So you can see that most actually, like we said yesterday, some of the signal of selection might be lost just because we cannot differentiate if it is because of spatial structure and genetic structure or only on the environment. But we still wanted to include both the RDA and the partial RDA, and we compared the outliers that we found. And well, we gave more weight to the outliers that were found by multiple gene environment association methods. And you can see that most of those that are in the intersection in the Venn diagram is related to PC2. So that's mostly summer air temperature, summer sea surface temperature, but also turbidity and salinity. And that's it. Just without going into details, you cannot see because of the bar at the bottom, but we've checked for gene close to those SNPs, and we find different biological function, cardiac muscles development, and immunity, the usual culprits. But we didn't go dive too deeply into that. So you've seen from this first population that at least in this study area, there's a lot of forces happening at the same time. There's isolation by distance. There's a lot of gene flow happening, mostly into neighboring rivers. There might be some isolation by colonization. So the order in which the regions were colonized might have a role to play in the genetic variation. And of course, we're pretty sure that there's also natural selection happening in this highly contrasted environment. And of course, there's a lot of limits to that part. So in the future, we want to expand our study area just to more represent the environment, because there's many more different environments where architecture evolves in the Arctic. We want to go for Nunavut, Northwest Territory, but also Greenland. And also we'll be moving on, and I'll be presenting some results on that on whole genome sequencing. So just having a higher density of marker. Want to include also other marker types than just SNPs. So structural variation that we might be able to identify. So inversion, insertions, and so on. And also we would like to apply those really interesting methods of genomic offset to kind of be able to say something about the future of adaptation in the Arctic. So I wanted to share a bit of newer results on phytogeography, mainly based on deglaciation. So I'll be focusing. We've increased our study area by a lot, but we're still trying to stay in the Arctic lineage that I presented earlier. So the blue points. We're trying to see if we can reconstruct the evolutionary history of this lineage and to see what are the impacts of this recent recolonization on the Arctic chart genome. Like I said, we've moved on to whole genome sequencing, but salmonids and Arctic char have like really huge genomes, so it's not really feasible to go for full out whole genome sequencing just because we want to represent a lot of populations. So we opted and still are trying to make it work low coverage whole genome sequencing. So rather than having like 10, 20x at each locus, you already have much more closer to 2 or 3. So you don't have a strict like genotype calling, but you're more working in a genotype likelihood. So you'll have, for example, 66% for that individuals in that SNP of being homozygous for that allele. So it changes a lot of the algorithm that we use. But still on the population level, we can kind of put all this information together and still yields a lot of information because we go from RADseq, for example, that focus on small parts of the genome to the bottom low coverage whole genome sequencing that covers much more of the genome. So this is our new study area that we still are expanding, and you can see how much west to east spatial structure is important in the principal component analysis. Half of the genetic variance is explained by that axis. Also found again our pattern of isolation by distance based on pairwise FSTs. And now to be able to say more about the evolutionary history of the region and how the range expansion since the last glaciation happened, we used a method based on the ancestral and derived allele, just because when the range is expanding, we are expecting it to be based on serial founders effect. So like you see in the diagram, a small number of individuals colonize the newly available habitat, and it can lead to higher drift. And so it can promote, it can accumulate derived alleles that were not found in more ancestral populations. So to identify those derived versus ancestral allele, we compare our data to whole genome data from other cousin species. So Atlantic salmon, species of Pacific salmon, and we have also the rainbow trout in there. So it's similar to the FST, but so you'll compare two populations, but you'll compare the amount of derived versus ancestral alleles. So if you have a positive directionality index, sorry for the there's no translation there. So if it's positive, you can infer that the range expansion happened from the population 1 to 2, and if it's negative, you actually expect the other way around. So we can compare all our pairs of populations, try to determine in which direction the range expansion should have happened. So this is what we did here. I represented the absolute value of psi with the boldness of the arrow, and just the colors represent the angle, so the direction. And you can see that most of it happened from the northwest to the southeast. And again, similar to isolation by distance that I showed with FST, but you can also see that if the more two populations are farther away, the more their directionality index is high, which is to be expected. And to go even further, we try to, from those information, infer the potential position of the expansion origin, aka the glacial refugium. So it's a method that's called time difference of arrival, and it's similar to what we think what we talk about triangulation. So when we are using, for example, a cellular service to triangulate the position of an apparel. So we will try to use the directionality index to see where is the most potential origin of that expansion. So that's what we did here. And it really fits our expectation. I was talking about Banks Island. This is, well, you can see in red the most probable position of the glacial refugium that I represented with the X, which is in the near vicinity of Banks Island. What I found interesting too is how the model kind of reaches zero before reaching the full extent of our study area. And you can see where it goes from yellow to white was a region that was really interesting, actually, because when I'm looking at the different index of genetic diversity, we really do see a clash between populations to the northwest of this limit and to the southeast, like you can see on the right. And it actually fits. So this is still preliminary data, and I don't know how much I can say about this, but this actually fits with the last ice barrier that separated the northwestern and the southeastern parts of the Arctic. So there might be something happening in that once this ice barrier disappear. Maybe there was a really rapid recolonization of the southeast. But yeah, we'll still have to dive deeper into that in the future. That's something that we want to do. So we would like to construct demographic models between the north and the south of the distribution, just to see maybe divergence time, and at which time, if we can pinpoint a timing of those recolonization. Of course, I talked just a bit about genetic diversity. So we saw that the northwestern part was less diverse than the southeastern part, which is actually the opposite of what we expected on the range expansion. So we want to also go deeper into that and also look at the mutation load of deleterious alleles to check if the recolonization might also have had an impact on the mutation loads of those populations. So on that, I want to thank you for your attention, and I'm really happy to answer any questions you have.
Okay, thanks, Xavier, for your really interesting talk. Is there any question from the audience? Okay, so, Francesco, please.
Okay. Do you have landlocked populations?
Yeah,
you do. Why? How come you didn't insert them in your data set?
Yeah, so that's a question I get a lot, and it's people have mostly focused on anadromous populations. Mainly, I would say, because it's mostly opportunistic sampling. So we partner a lot with local fishermen, and people are just not that interested to fish for landlocked fish. But definitely there has been some studies before that compare, like in the same lake, the fish that go to the sea versus the fish that stays. Are they the same populations? Is there gene flow? Is there separation? And so it's still an area where there's some work to be done, but generally there is quite a lot of gene flow, and the life history traits are not that clean cut either. I've heard tales of a fish when you look at the otoliths and you see like what is the age of the first migration to sea. Some fish live for 18 years in the lake before going for the first time in the sea. So to come back to your question, we focus mostly on anadromous just because that's the sample that we can get without.
Okay, and one more very, very fast.
Yeah.
Do you have any idea if there are different run timings in the same river, like a group starting on August and maybe another one in December, like salmon do?
To my knowledge, it's not as so. We have like some species of salmon that really like have a phenotype that is like the spring run and the fall run and the late run and early run. To my knowledge, there's no such knowledge of really like separate like populations of separate genetic background that migrate at different times. But there might be something. Some studies suggest that the females go back to the river before the males, or some things like that.
Okay.
But,
Thanks.
Okay, I also have a, I think, fast question. Is a more technical aspect. So you went for GBS as a genotyping technique, but according to my knowledge, there is a SNP chip, a commercial SNP chip available for the species. So is there any particular reason why you decided to use GBS?
Actually, at that time, I'm not sure if the SNP chip was already available when we chose the GBS. Actually, we first wanted to use Rapture, which is like RAD, but you actually have primers that capture specific SNPs that you identified before, and we really had too much challenges in the lab. It was not working at all, so we switched to GBS to get the work done.
Okay,
and now
thanks.
we switched to whole genome sequencing.
Questions? So
Thank you very much. No, I had a question close to Francesco ask you. First of all, thank you very much for your presentation. I would like just to say if you find if you already find some genes linked to the migration phenomena, because on 2020 was described on Science that only a short region in the whole genome it's linked to the migration phenomena and the timing.
So I
did
think
you find
that's in a species of salmon that
yeah there was a salmon.
Yeah, so no, we haven't looked directly at those. We haven't like sequenced specifically those region, and from at least our GBS data, there was no SNP like in that region that popped up. But that's GBS. You do not cover necessarily like the full extent of. But that's something, yeah. Now that we have whole genome sequencing, maybe we want to go deeper in that's the candidate genes that we know in salmonids that are found like in multiple species of salmonids too. So yeah, something for the future.
Yeah. Thank you.
okay, so thanks again, Xavier.
Now we have the second presentation by Ivana Maguire. I hope I said it right.
Perfectly.
From Croatia, with a presentation entitled Geographical Distribution of the Native Crayfish Genetic Diversity in Croatia.
Thank you for introduction. We are back to Europe, into the neighborhood. And I have to say, I'm really glad I'm here. It really is an inspiring workshop. I got many ideas, and in this presentation, I will actually give a tiny little bit of the work we are doing on freshwater crayfish in Croatia. So for the beginning, even though I know majority of you knows, Croatia is situated in Southeast Europe on the Balkan Peninsula, western part. That is actually known as one of the glacial refugia in Europe. Actually, thanks to its position, it encompasses 4 different biogeographical regions. So on the north, we have Pannonian region, then we have continental biogeographical region, Alpine, and Mediterranean biogeographical region. Thanks to all those geological events that happen in the evolutionary past, help actually development of really rich hydrological network in Croatia, with rivers belonging to both Black Sea drainage and Adriatic Sea drainage. And again, being in the position it is that rich hydrology enable evolution of numerous freshwater taxa, among others also freshwater crayfish. So within Croatia, we have 4 native freshwater crayfish species. We have noble crayfish, or Astacus astacus, that is distributed mainly in continental part of Croatia, but also some populations were introduced to Mediterranean region, because Astacus astacus, a noble crayfish, is a species that people use in their consumption. So it was frequently take around and introduced in many places. Second species is Pontastacus leptodactylus, or narrow claw crayfish, that is distributed in lowland Croatia in bigger rivers. This is quite robust species that can actually stand quite polluted waters. So it shows some signs of spreading and pushing out noble crayfish. Then we have Austropotamobius torrentium, or stone crayfish, the smallest of all native European crayfish species. In Croatia, it comes in smaller stream on higher altitudes. So mainly it is present in alpine geographical region, but also in some of the mountains in continental part. And there are few rivers in Mediterranean Croatia, in streams that are draining Adriatic Sea, where I suppose they stay trapped after the mountains rise. And finally, we have white claw crayfish, or Austropotamobius palipes, complex species. I'll say something about it later. This species is distributed only in the water bodies draining Adriatic Sea. Again, as with numerous freshwater taxa, there are many pressures onto populations, and the numbers are declining. So it's all those anthropogenic pressures from alteration of the habitat, pollution. Then on top of that, we have really extreme climatic conditions with really prolonged drought periods that actually cause drastic negative influence on crayfish population. And finally, we have also invasive alien crayfish species that are spreading, and that, apart from being more aggressive, more fertile, push out or displace native crayfish due to the pathogens that they bring or brought into Europe with them. Since I'm working in freshwater crayfish studies for many years, I managed to collect the data, recent data, and well, I have some from the past, and those actually enable me to calculate or estimate how many percentages of populations were lost due to those previously mentioned reasons. So as you can see, or maybe not completely, according to those estimations, 55% of populations of noble crayfish disappear, 29% of stone crayfish, and almost 70% of originally existing white claw crayfish population disappeared. This actually was kind of starting point to use IUCN methodology to be able to categorize populations or species that do exist in Croatia. So for Astacus astacus, a noble crayfish, it proposed national categorization that it is a vulnerable species. Palipes, or white claw crayfish, is endangered, and actually stone crayfish is vulnerable. The fourth species, as I said, narrow claw crayfish, is quite robust, and it is actually least concern. Of course, because of all of that, all three mentioned previously species are protected, and they are protected by national legislation in Croatia, but also they are protected globally in Europe by Habitat Directive and Berne Convention. So this brings us to the aim of this presentation. I said it is simple, it's just a tiny little part of the things that we've been doing on freshwater crayfish. So the aim, simply to put it, were to assess genetic diversity of the native crayfish species in Croatia freshwaters using DNA barcoding, and also to display that diversity in the space, or basically to show them on geographical maps. So what we did actually in this presentation, I will show the results for more than 730 individuals. We, of course, sample tissue, crayfish tissue, and after DNA extraction, we amplify barcode fragment. After that, and after sequencing, we prepared a barcode database from which we then pull a number of haplotypes per population that I will show later. On the other side, at the same time, we, of course, recorded coordinates of each population that we sampled. And then actually we took our gadget into the lab and using ArcGIS program package, we prepare layer with distribution of populations. And finally, what I will show is that we overlap cytochrome oxidase haplotypes with the map of their distribution within Croatia. That brings us to the results. Altogether, we collected or we obtained 525 cytochrome oxidase sequences. And in this table, you can see the total number of cytochrome oxidase haplotypes and, of course, number of unique cytochrome oxidase haplotypes per each species. So as you can see, they range from 16 for noble crayfish to 45 for stone crayfish. To give more details on those numbers, I have to, I suppose, give few more details about each species and what we actually know about them. So first, here are some previously published results on stone crayfish. What is important to know that within the Europe, there are 9 divergent lineages of stone crayfish. And as you can probably see from this map, within Croatia, we have 7 of them. And they are actually on this network encircled with red circles. The highest diversity of Austropotamobius torrentium was revealed in alpine biogeographical region, actually in dinaric karstic habitats. When we show the number of haplotypes per population per lineage, and actually those lineages we treat as evolutionary significant units. So here is the map with the number of recorded different haplotypes within each population. And the number of haplotypes per population range from 1 to 7. Second species is Austropotamobius polypes species complex. Taxonomy of this complex is still, I would say, under the discussion. And some people say there are 2 species within it. Some people say there are different lineages. But as far as I know, the last comprehensive study on the phylogenetic relationships and on the phylogeography of the white claw crayfish was done by us in 2016. And actually what we wanted to do is to fill the gap that was missing at that time with the population from Croatia. And possibly you can see from this map that within Croatia we have 2 lineages of putative subspecies, Austropotamobius italicus carsicus and meridionalis. The same thing shown on this network. So 2 different groups of white claw crayfish. The highest diversity, of course, for this species within Croatia was revealed in the Mediterranean biogeographical region, precisely in Istra or Istrian Peninsula. When we want to see number of haplotypes per population, I will show them on this slide. There are some dots and there are some triangles. So for triangles, we actually did analysis and we have number of haplotypes. Since Croatian coast is long, not as long as the Italian one, I divided it into parts. So this is the situation in Northern Adriatic. And this is the situation in Middle and Southern Adriatic, and the number of haplotypes per population in studied populations vary or range from 1 to 4. Then we come to the Astacus astacus, or the noble crayfish. Within Europe, there are like 6 lineages described. And on this slide, you can see results for Croatia. As you can see, within Croatia, we have 2 previously described lineages, plus we, in our last study, actually described or discovered a new one. The highest diversity for this species was revealed in continental biogeographical region, and actually number of haplotypes per population per lineage again is shown in this slide, and it ranged from 1 to 3 per population. And finally, the last species, narrow claw crayfish, or Pontastacus leptodactylus. I would dare saying that study we did long time ago, back in 2012 or 14, I'm not sure, was the first actually that tried to put into one place and find something about phylogenetic relationship within this. Actually, it's also complex. So that study from long time ago actually show us that within sampled narrow claw crayfish, there are 2 phylogenetic groups or 2 lineages, one that is typical for European populations and one that exists in Asia. As you can see again with the red color, I put circles around haplotypes or populations that are recorded in Croatia, but you can also see that there are some Asian haplotypes or some of the, one population actually that, show Asian origin. So I have to explain here that this is a consequence of the fact that at that time it was possible to buy live narrow claw crayfish in Zagreb on the market, on fish market, and actually they were introduced from Armenia. So somebody obviously felt pity for them and put them into one artificial lake in one park in Zagreb, and that's where we found them. And that's how they actually, one of those rare occasions when you have illegal introduction and you can somehow connect it to places and people. So this is actually a map showing number of haplotypes per population, and as you can see, the number range from 1 to 13, and 13 is actually number of haplotypes in that one lake where narrow claw crayfish were stacked from market originating from Armenia. So this is the end, and now I will share with you some conclusions. First, definitely Croatian populations harbor an important part of species genetic diversity, and I would say represent significant genetic reservoir, not just on Croatian, but also European level. I already mentioned it, but I will repeat it. Stone crayfish highest diversity was revealed in Alpine biogeographical region, while continental biogeographical region hosts the highest diversity of noble crayfish, and Mediterranean biogeographical region of the white claw crayfish. This simple way is good way to actually, in combination, geographical distribution of diversity can give us some ideas, and it can be used as a useful tool for guiding conservation of endangered species. Of course, we heard here many results and many ideas and suggestions how deep we can go, and we are aware of that. We know that obtained result will serve and already served in combination with population genetics data and species distribution modeling as a basis for conservation of these endangered crayfish species. I said they already served, so we have already some results, and I can show them. This is an example for noble crayfish in which we apply species distribution modeling, in which we use some scenarios for the future under different, climatic change scenarios. And this one show us in gray the parts or habitats that may become unsuitable for noble crayfish. And as you can see, this eastern part of Croatia actually in 2070 under a pessimistic scenario, but situation is quite similar for that less pessimistic scenario. They will probably disappear. And if we would count all the populations and those regions, we actually estimated that possibly 50% of cytochrome oxidase haplotypes recorded currently in Croatia will be potentially lost. We did similar thing for the stone crayfish. And again, as you can see from this map, different symbols actually present different evolutionary significant units. And orange background actually presents habitats that are predicted to be lost again in 2070 under the, well, that pessimistic scenario. According to this model, 44% of population with highest or unique genetic diversity are located in areas that are predicted to be lost, and actually that includes 3 evolutionary significant lineage. So future is not so bright, but we are working on some solutions and some ways to help them to survive, because they are keystone species and they're really important, not only for the health of the fresh waters, but also at the end for all of us. So thank you for your attention, and grazie.
Is there any question? No? Okay, I have one for you. And
Hope not hard.
no, just a curiosity. Did you ever find a Torrentium and Polypes in sympatry?
No.
Never happened.
They do, Polypes normally comes in a bigger rivers, and Torrentium gets stuck in some tiny little streams connected to that bigger river, but never. I found in few occasions noble crayfish with stone crayfish, but this was consequence of people introducing
the introduction.
Noble crayfish, and eventually Torrentium stone crayfish push noble crayfish out.
I see. Okay, thank you. Any other question?
It was so clear and so simple. There are no questions.
I have one.
Huh?
I have one before you leave. Okay, so I noticed that when you compared the number of haplotypes you found in Torrentium and Polypes, the range, okay, the values of the range were quite different. So I was wondering if that was mirroring perhaps a difference in sampling size, or if it's an actual feature of how diversity occurs and is distributed.
Well, there is. I mean, the sample size wasn't always the same, of course, and I didn't do any kind of normalization. It normally goes 5 crayfish and more. But I think it is due to the fact that actually what we catch nowadays on the coast, white claw crayfish, lots of it's already gone. And stone crayfish, it's in more remote places, more isolated. And it is, I think, hotspot of diversity on European level. So we still have quite a bit. So I think that would explain the.
Thanks a lot. Okay.
Other questions? Maybe I have one more. We have time. May I?
Okay. And who's got the question? You, sorry.
Just a fast one. We wrote a paper on Polypes with a separation in Italy between northern Italy and central Italy. And since Croatia should belong to the Po River geography, right? Yes. A connection. Did you try to match your haplotypes with the northern Italian haplotypes?
Yes, it is actually in this paper. We actually showed that some at the same time on Croatian side and Italian side. I think it's Marca region. Marca.
Right. Yeah, that's a border.
Yes, yeah. And I don't know by heart, but there are like details in that paper, and I can check if.
All right. Thank you.
Okay, so we move on to the next speaker. So I call Romolo Caniglia from the Italian Institute for Environmental Protection and Research, ISPRA, to take floor. and to present his contribution on conservation genomics and anthropogenic hybridization through a number of case studies on Italian wildcat, wild boar, and wolf.
Good afternoon, everybody. Thanks a lot to all the staff for the organization of this very interesting but also very useful workshop. And thanks a lot for inviting our team to participate. In this presentation, I will show you a multi-level genomic approach we applied to answer challenging and partially new evolutionary questions about the Italian wildcat, wild boar, and wolf populations, which are three taxa very interesting from both the conservation and the management points of view, and which are all currently potentially threatened by anthropogenic hybridization. Just to recap, hybridization can be generally defined as the cross between individuals belonging to two different species, subspecies, or even populations, distinguishable at least for one hereditary character. Natural hybridization is a very important evolutionary process since it can contribute to the birth of new taxa. Unfortunately, during the last decades, a new kind of hybridization is spreading, the so-called anthropogenic hybridization, which is caused or favored directly or indirectly by human activities, and which can have deleterious consequences on the fitness of natural populations whose genomes can be compromised or even extinct by the introgression of alien or domestic genes. Before going on with the presentation, it's important to clarify some concepts to avoid erroneous conservation management actions. For example, the term F1s indicates individuals directly originated from the crossing between individuals from two taxa. If these F1s are fertile, they can cross with individuals belonging to one of the parental populations, originating the so-called fourth generation of backcrosses. And if backcrosses with the individuals of one of the parental populations go on, they can start the phenomenon of the gene introgression, which implies the incorporation of genes or genomic variants from one taxon to another. Identifying hybrids, fourth generation recent hybrids, can be relatively simple because they theoretically share half of the genomic components of one parental population and half from the other. Unfortunately, the identification of ancient hybrids can be demanding or virtually impossible because of the dilution of the genomic component of the population not involved in the crossings. So it is necessary to define reliable criteria to reliably distinguish pure from admixed individuals. For example, studying the threat of the anthropogenic hybridization in the Italian wolf population, we recently developed and applied a standardized workflow based on the use of well-defined reference dog and wolf populations and appropriate assignment thresholds aimed at minimizing the risk to erroneously classify a wolf as a hybrid and vice versa. The detection of hybrids can be based on the application, for example, of anomalous morphological or behavioral characters. However, they can be subjective or not quantifiable. Therefore, more reliable results in hybrid identification can be obtained using molecular tools. For example, the use of uniparental markers can provide the directionality of the hybridization, whereas the use of a few dozens of biparental markers can allow the reconstruction of genetic profiles of recent hybrids, which can be identified comparing them with reference populations, for example, in multivariate analysis or in Bayesian clustering procedures. During the last decades, the threat of anthropogenic hybridization has been extensively investigated in the Italian wildcat, wild boar, and wolf populations, as demonstrated by the high number of genetic studies published. However, most of these studies were mainly based on a limited number of molecular markers, so they provided only partial information. Likely, during the genomic era we are experiencing, it is possible the genotyping of dozens of biological samples at hundreds, at thousands of molecular markers, or even at entire genomes, allowing to obtain very detailed, extremely detailed information. For example, the identification of hybrids very ancient, but also the identification, the detection of ancient population gene flow, the identification of deep signals of admixture chromosome by chromosome, and even the identification of the genetic basis of some anomalous phenotypes, which can be considered putative indicator of hybridization. For this reason, in our study, we applied genomic tools to better understand and clarify if anthropogenic hybridization is really widespread and if it is really affecting the long-term survival of the Italian wildcat, wild boar, and wolf populations, which are taxa which experienced very similar evolutionary scenarios, characterized by protracted isolation, recurrent bottlenecks, habitat loss or fragmentation, and recent re-expansion in human-dominated landscapes with a high density of their feral domestic counterparts. Using specific SNP chip microarrays, we genotyped more than 50 felids at more than 63,000 SNPs, more than 150 pigs at more than 60,000 SNPs, and more than 200 canids at more than 170,000 SNPs. Additionally, in the wolf case study, we analyzed even 6 whole genomes obtained with a very high average coverage. Multilocus genotypes were used to carry out multivariate, Bayesian, and maximum likelihood analysis to investigate population structure and for a preliminary hybrid identification. Additionally, local ancestry reconstruction methods were applied to investigate significance, directionality, and the timing of the admixture. And finally, GWAS and gene ontology methods were applied to detect the genetic basis of anomalous phenotypes in putative hybrids. In the wildcat case study, traditional multivariate and assignment analysis allowed us to better clarify the overall European wildcat population structure and substructure. They allowed to identify admixed individuals date back from the 6th to 22nd generation in the past, and gene ontology methods allowed us to clearly distinguish in the admixed individuals domestic-like genes mainly related to cognition and neural crest development from wild-like genes mainly related to social behavior, adaptive and metabolic processes. In the wild boar case study, traditional assignment and multivariate methods clearly confirmed that the investigate Italian wild boar populations were highly differentiated from all the other wild boar European populations analyzed. In particular, the most differentiated Italian populations resulted to be the Sardinian, the Maremma, and the Castelporziano presidential estate populations, which can be considered the result of a long-lasting isolation. Surprisingly, we didn't find significant domestic ancestry in the Italian wild boar populations, but conversely, we identified significant wild ancestry components in some local pig breeds. The maximum likelihood 3 obtained using the innovative approach implemented in the software TreeMix clearly confirmed the overall population structure at the European level, and additionally allowed us to detect some migration significant signals, for example, from the central Italian wild boar populations toward the Sardinia populations, and from the population of Maremma and Castelporziano towards the population living in Calabria. In the wolf case study, traditional multivariate and assignment procedures combined with the local ancestry analysis allowed us to identify hybrids up to the 19th generation before sampling, and definitively clarify that for the Italian wolf population, most of the hybridization events occurred not only during the 70s, which corresponds to the last bottleneck, but they happened mostly during the 80s, during the phase of population re-expansion. GWAS and gene ontology methods allowed us to identify clearly the genetic basis of 3 anomalous phenotypes in the Italian wolf populations, detecting highly associated signals for the black coat, for the white claws, and multiple associated signals for the spore. The associated haplotypes to these three anomalous phenotypes confirmed to be clearly dog-derived. However, we cannot consider them direct indicators of hybridization because they were found mostly in individuals date back as admixed to the fifth generation in the past. The analysis of entire genomes from the Southern European wolf populations clearly showed that the present-day Eurasian wolf populations share a common ancestor date back to about 36,000 years ago, and also clearly show that the current isolated Iberian, Italian, and Dinaric wolf populations started to diverge about 11,000 years ago. Finally, exploiting the availability of many genomic profiles of dogs, jackals, Italian, Iberian, and Dinaric wolves, we selected and optimized a reduced panel of SNPs, which can be successfully applied to reliably distinguish natural population gene flow from anthropogenic hybridization up to the first generation of the crossing, both analyzing invisibly and non-invisibly collected samples. So, our multi-species genomic study clearly shows that multi-level genome-wide local ancestry reconstruction methods can be successfully applied to better investigate population structure, substructure, and dynamics, but also to assess individual domestic ancestry proportions, to clarify admixture patterns, even in deeply introgressed populations, and especially to identify candidate genes or genomic regions responsible for anomalous phenotypes, potentially indicators of hybridization. And all this information can be combined and can be used to plan and support sound conservation management actions. Future landscape genomic approaches combining genome-wide data and environmental variables could surely help researchers to better understand if and how anthropic or natural barriers may shape genome-wide variations, and even if environmental variables, specific local environmental variables, can favor or not the selection or the adaptation of particular locally adapted genes in these three fascinating Italian mammal species. Thanks for your attention, and I'm here for any questions or suggestions.
Thanks, Romolo, for your comprehensive presentation and for how you treated the topic of hybridization, which is quite relevant for conservation action. So is there any question from the audience? Paolo, yeah.
I have an observation or a suggestion. As many of the talks I have heard and or I read the abstract are dealing with adaptation, that I think it is an extremely important issue in wildlife conservation management and so on. And adaptation is tightly linked to genomics on one side, but on epigenomics on the other. Okay, probably the first response of a species or a population is epigenetic rather than genetics. And now there is this Luma technology. I don't know if you heard about it. It is a low-cost technology that is able to evaluate the overall methylation status of the different population. We are testing it in another project on livestock, but maybe it can be some. I don't know if there is already data available in wildlife, and if can be useful also for this kind of investigations.
Yes, very interesting reflection. And yes, the next future steps will be to apply analysis on whole genomes to test for local adaptation, selection of particular traits, and probably we will move on transcriptomes and epigenomics. However, it is a challenging topic because dealing with threatened taxa, living taxa, it is difficult to obtain really fresh samples from living animals. But this is a future challenge, and we are going to apply. We are going to apply especially transcriptome analysis, and we had an experience with a team of colleagues who analyzed epigenomic patterns in some wolf-dog hybrids or canid admixed individuals, but they were able to do that analyzing individuals captive bred. So we are evaluating, and it is possible, and it is a future possible step to do.
Other questions?
Yeah, I have one very fast. When you speak about hybrids, can you identify if the dog is like a shepherd or a guard dog or the strain?
Yes, we were lucky because the Illumina SNP chip we applied in the wolf-dog hybridization detection was particularly developed to distinguish dog breeds. And from a preliminary analysis, the breed which mostly contributed to the admixture for the Italian population was the shepherd dog. But probably because during the 80s, the period during which most of the hybridization events occurred, that was the most widespread breed in Italy. But with the friend and colleague Paola Crepaldi, we are going to plan to investigate such a topic in a deeper way.
Okay, and that was good because you don't see it from the phenotype, right? You see it less.
Yeah.
It's a joke.
Yes. It's very difficult because we detected phenotype morphologically wolves, but genetically admixed individuals, and vice versa. So there is not so a direct link between genotype and phenotypes. And probably analyzing whole genomes, we could use RATAC methods to develop specific SNP panels to address such questions.
Okay. Other questions? No, so thank you.
Thank you to you.
Thank you very much. Well, Licia, it's your turn for the workshop closure.
Okay, so I will be very brief, but okay, once more, let me thank all the people who contributed to this workshop, the chairman, the main lecturers, and all the speakers, because I think that all together, the contribution allowed us to walk and move across the extension of this topic in a very nice and effective way, starting from the very beginning, the birth of the landscape genomics and related approaches, up to the applications, a wide number of applications in the field. And of course, I want to thank you for attending the workshop, because it was very nice having the chance to meet so many attendees in person again after, you know, the pandemics that hit all of us. So, so here, I want to acknowledge the contribution of the WIGGIS organizing and scientific committees, which during the last months worked in the background to make this event possible. And again, of course, the EU LIFE framework for making the funds available for organizing this event. And these are further acknowledgements to also the partners of Like Law projects overall. And a special thanks from my side is to the people of my group, Elisa, Maria Antonietta, Matilde, and Julia, who during these days worked in the background to support the evolution of this event. Okay, so thanks again, everyone.